Hi everyone, this video will show you the actual experimental steps and data collected for the qualitative analysis for Group I Cations lab. Your instructor may ask you questions related to the procedure or data shown in the video. As you're going through the videos, it is useful to have the lab procedure and lab report with you so you can write down all the relevant observations. The experiment will be carried out with one sample of all the known cations, which we call the control group, and then two samples containing unknown Group I cations. The two samples are labeled 1E and 7T on the test tubes. Your instructor may assign you to identify which cations are present in one or both of the unknowns. The experiment starts with the addition of 6 molar HCl to all the three samples. The samples are then centrifuged or spun at a high rate to separate the solid particles from the aqueous solution. Because of the high speed, each test tube must have another one with the same weight placed across from it to balance. Here are the three samples: control, 1E, and 7T after centrifugation. We then add another drop of HCl to each sample and centrifuge it again. After this second centrifugation, the supernatant or solution in each test tube is decanted into a waste container. We then wash the precipitate with water to remove any contaminants that may be trapped in the precipitate. This is done by adding water, stirring the contents of the test tube, and centrifuging. The supernatant is again decanted into waste. Afterwards, we add about 2 milliliters of deionized water into each test tube and place the test tube in a hot water bath. While they're heating, we stir the content of each test tube with a stirring rod. After 3 to 4 minutes of heating, the hot test tubes were again centrifuged. The hot supernatant is decanted into a new, clean test tube. To this solution, we add 6 molar acetic acid, followed by 1 molar potassium chromate. The results are shown here for the control, 1E, and 7T. Next, we went back to the precipitate that stays in the test tube after heating. To it, we add 6 molar ammonia and mix the content with a stirring rod. We then centrifuge the samples. This is what the positive control and sample 1E look like. Sample 7T just gives a clear solution. We decant the supernatant into 3 clean test tubes. To each, we add enough 6 molar nitric acid to make the solution acidic by litmus. Both the control and sample 7T have this appearance. This marks the end of this experiment.